The overall goal of this microscopy protocol is to measure in vivo plant calcium dynamics as insects feed from a leaf.
This method can help answer key questions about plant-aphid interactions, such as how plants detect and respond to aphid pests. The main advantage of this technique is that it allows us to detect plant calcium signals in vivo whilst a live insect is feeding from the leaf. Although this method can provide an insight into the role of calcium signaling during aphid feeding, it can also be applied to other systems, such as new insects or plants, or even to detect signals in response to abiotic stresses, such as salt.
Begin by growing 35 SG Camp 3 Arabidopsis plants, and on day 11, rearing aphids as described in the text protocol. On day 19 of the experiment, remove the seedlings from the controlled environment room. Detach the largest leaf from each plant using a pair of sharp scissors. Next, use a pair of tweezers to place each leaf abaxial surface facing up into separate wells of a 96-well plate containing 300 microliters of distilled water per well. To reduce detachment stress in the leaves, cover the plate with clear plastic wrap and aluminum foil and leave it at room temperature overnight. On day 20, collect the aged aphids from the colony established on day 11 by moving the infested plant from the soil into a transparent box with a lid. Make sure to keep the insects contained in the box during the experiment. Remove the aluminum foil from the 96-well plate